Thank you very much, Seraphine, for agreeing to give a Build a Cell seminar, and we can get started.
Yes, thank you, Kate, for having me. It's really a great pleasure to be part of the seminar series I'm a big fan of, and I regularly follow you also on YouTube in the seminar series. Yeah, so my name is Seraphine Wegner, and by training, I'm a chemist. And I'm very much excited about this idea of synthesizing a cell, because in chemistry, I think many synthetic targets have already been achieved, but synthesizing a cell from the bottom up is really the ultimate goal. So yeah, I'm at the University of Münster in the northwest of Germany at the medical school, but by training, I'm a chemist, as you will also see from my talk. So yeah, in my group, we really approach synthetic biology from the bottom up, really have this perspective of a chemist of synthesizing things. But this idea of using a modular approach, this Lego approach that to biology is of course very appealing for that. This is something that an approach that we have in chemistry over and over again, where we define functional groups and understand how they react and interact with each other. And it's exciting to take this perspective also into biology. So we do this bottom-up synthetic biology on two scales. The first one, which I think also this crowd is very familiar with, is to use molecular building blocks and synthesize synthetic cells by controlling how these molecules interact with each other. And by assembling molecules that come from biology, but also could be of synthetic origin, we can build cell mimetic systems that replicate certain cell functions that we associate with light. And by understanding how these building blocks work together and give rise to function, we can hope to engineer these cell mimetic systems and maybe one day really realize the big bang of chemistry and biology and transform non-living molecules into living ones. Another thing that we do in my group that I'm not going to focus too much on is to synthesize tissues from the bottom up. And here we start off with cells as building blocks and assemble these into multicellular assemblies, microtissues if we use mammalian cells, but we also have similar approaches with bacteria, and we try to build up biofilms. And in marrying also synthetic and living cells, we hope to also produce hybrid systems that could have enhanced capabilities. In all of these approaches, a very crucial element is to control when and where these interactions are taking place. And therefore, an overarching theme in my group is to regulate these systems with light, because we believe that light is the best way of regulating a system. First of all, you have super high temporal control, because you can deliver light at the speed of light, and that's no exaggeration. And you can also remove it by simply turning off the light at the speed of light. And I believe that there is no other input that could give you such temporal control. The other big advantage is, of course, the high spatial control that you get. You can focus a beam of light very precisely. That's exactly what your screen is currently doing. In each pixel, you have a defined light intensity, a defined gray value, and you can project any pattern that you would like on the screen. And if you use a confocal microscope, this spot can be down to a diffraction limited spot. Yet this is still very scalable, and you can use photomasks or just simple LEDs and project patterns on your sample. At the same time, light is also a very tunable stimulus, because you can play with light intensity or illumination patterns and thereby titrate your system. The other thing that we like about light is that the rainbow has many colors, and therefore we can use different colors of light to address different functions within our systems orthogonally. And to do so, we use a set of wonderful proteins that come from optogenetics. These are photoswitchable proteins that respond to visible light. And from a chemical standpoint of view, most of these proteins that we use are light-dependent dimerizers. We have proteins that heterodimerize under light, or proteins that homodimerize under light. So this allows us to bring in together different or the same entities. Another great feature of these proteins is that most of these interactions are reversible in the dark. And this allows you to then introduce dynamics into the system. These proteins respond to blue or red light, and the red light response are ones you can even deactivate active by illuminating with far red light. And it should also be noted that these proteins have already evolved to work in very complex biological environments. So these protein-protein interactions are highly specific, and these proteins respond to very low intensities of visible light that do not cause any adverse reactions in your system, because these proteins in the end come from photosynthetic organisms that have evolved to operate with light that is available on our planet. And so in my group, we believe that these photoswitchable proteins should definitely be part of this toolbox to build synthetic cells, because a lot of functions arise from spatial temporal control. If you think about, there's many examples, but a few would be cell migration. Cell migration works because in the front and in the back of the cell, there's different chemistries that are happening. At another scale, at the tissue scale, the organization of cells and when and where these cells interact determines how they function as a tissue, as a community, and also how they communicate with each other. And that's why we are introducing photoswitchable proteins to mimic these and many other processes that arise from spatial temporal control. To maybe give you a first example, I would like to look here. You see a migrating cell, and in this migrating cell, the adhesions have been labeled with a fluorescent marker. And you can appreciate as the cell moves forward, there's new adhesions that form in the front, while other adhesions disassemble in the back. And while these adhesions are controlled in space and time, they are still reversible. There is a matching dynamic between the back and the front of the cells, and this asymmetry in adhesions is maintained at the length scale of a single cell. And so we asked the question, can we replicate this in a completely synthetic system by replacing basically the cell with a giant unilamellar vesicle, so a lipid membrane basically filled with buffer, and functionalize the surface of these cells with artificial adhesion receptors. So this is the protein. We use the one protein called micro, and micro can bind to the protein islet under blue light illumination, and thereby we intended to initiate adhesions of these vesicles. This was the design, and here's how it looks from the side. So in red, you see the vesicle, the membrane, and in green, you see the reflection of the glass light that it's sitting on. And you can appreciate that if you illuminate with blue light, you see an increase in adhesion area and the vesicle gets deformed and squished down. And then when you stop illumination, you can appreciate that these adhesions reverse and the vesicle detaches. So this shows you the temporal control. And now we add spatial control to this. And here we are looking at the vesicles from the bottom. And you can see that this lower vesicle moves into the illuminated area because we are triggering the adhesions just in that half of the vesicle. And by moving this illuminated area forward, we can basically control where this vesicle attaches and recapitulate this asymmetry in adhesions that we also see in a living cell. And we see that this allows the cell to move forward. So what else? The cells are, of course, not in isolation. They perceive many different inputs, amongst others light, but also they interact with many other players in their surroundings. So when we think about cells in their environment and in communities, we have to really think about how these communities are structures, what type of signals they will exchange, when and where they will exchange this, and what type of behavior can arise from this. So to address some of these questions, we have replicated this phenomenon in synthetic cells. So one important decision that a cell has to take is where it will live. Will it colonize it or stay in a certain place, or will it leave because it doesn't fit its needs? And to make these type of decisions, cells actually have to integrate many different signals in their environment and see where they want to stay. And so these inputs can be light, but also redox or the availability of certain metal ions or pH, for example. And to replicate this, we again use our adhesion unit that I just introduced to you and wanted to control the adhesion of a synthetic cell with multiple inputs. So to introduce light sensitivity, I guess I already showed you this, we use photoswitchable proteins so that we can trigger adhesions under light. But at the same time, it should be noted that this photoswitchable protein islet is a love domain protein. And so it's actually not just sensitive to light, but also to redox. And when we oxidize it, then this interaction no longer takes place. So this is a way of then making also the system redox sensitive. Moreover, we also can use our functionalization chemistry to introduce further sensitivities. And in most of our work, we use the interaction between the his tag of our protein and NTA functionalities to immobilize these proteins on the surfaces of our synthetic cells. Well, these interactions are actually sensitive to metal ions because they need this mediator ion in the center for this interaction to work. And they're also sensitive to pH because at low pH, these ligands get deprotonated and the complex falls apart. And so thereby, by combining this his-tag nickel-NTA interaction with our photoswitchable islet nanoprotein-protein interaction, we designed an adhesion unit that is sensitive to all these 4 different inputs. So that was the design. And here is how we realized it. So in the first iteration, we used a polystyrene bead and immobilized our photoswitchable protein islet to a nickel-NTA interaction and looked at the adhesion to a functionalized glass slide that has nano on top. So you can appreciate that under light, at neutral pH, under reducing conditions, and in the presence of the metal ion, we see many of these polystyrene beads on the surface. But if we do not satisfy one of these conditions, then you see that very few of these beads adhere on the surface. So if we keep the system in the dark, if we lower the pH to pH 3.5, if we introduce an oxidative stress in the form of hydrogen peroxide, or remove the metal ion, then this adhesion no longer works. And it's not just one by one, but you can look at all 16 combinations and you can appreciate that you have a quadruple end gate and all 4 of these parameters have to be met for these adhesions to work. So what could we use this for? And here we collaborated with another group who had developed a light to ATP conversion module in a synthetic cell. And in these synthetic cells, you have a bacterial rhodopsin that uses light as a source of energy to pump protons across the membrane. And then within the same vesicle, there's an ATP synthase implemented. And this ATP synthase can then use this pH gradient to convert ADP to ATP. And if you look at the requirements of this light to ATP conversion module, you will also appreciate that it needs light, of course, to power the bacterial rhodopsin. It operates only at neutral pH, and acidic pH does disturb it because it cannot establish the pH gradient it requires. It requires reducing environment as oxidation causes the oxidation of some key cysteines in these proteins. And it requires the presence of metal ions as these proteins also have metal ion cofactors. So you can see that it has the same requirements as our adhesion module. And so we then combined the two to see if we can get a synthetic cell that knows where to adhere so that it can produce its ATP. So if we first look at the adhesion module, we can see that these vesicles bind to a surface under light, which we can see in a QCMD experiment. And you can see the adhesion of these vesicles to a decrease in the frequency and at the same time an increase in the dissipation, which basically means that there is large wobbly objects that bound to the surface. And conversely, in the dark, you see that the frequency only decreases slightly, and at the same time, also the dissipation only goes up a little. So we have these vesicles are able to bind to these surfaces under light and where they can also should be able to synthesize ATP, and this we can confirm by analyzing the ATP production on these surfaces, and we see substantially higher ATP production under blue light than in the dark. And so we here have a synthetic cell that basically can take multiple inputs from its environment, can process them through chemical functionalities, and then respond to a combination of these. This is nice. So here the cell is by itself, but we want to go to communities. And so we have cells that can function as senders, and then there is receivers. The senders send out a signal and the receivers should respond to it. Yet this communication actually depends on many other things. For example, the distance between the sender and the receiver. If the distance is large, you need a lot of signal, and as synthetic cells usually are limited in their capacities, signaling actually works more efficiently if they are in close proximity. And so we replicated this in synthetic systems. So now we have here giant unilamellar vesicles, and one functions as a sender, and it has calcium as a signal loaded into it. The receiver has a calcium-sensitive dye called Rod2, and if we then can release this calcium signal by adding an ionophore. Yet, as I mentioned, if the distance is too far, then the signal dilutes into the environment before it can reach the receiver. Yet, if we could bring these two in close proximity, then the sender would be able to perceive the signal, and that would be visible then as an increase in fluorescence. And to regulate this communication, we implemented our photoswitchable adhesion molecules on the surfaces of these so that we could trigger adhesions in the blue light and bring them in proximity. So that was the design. And here is how it looks under the microscope. So the sender UVs will always be labeled with a green fluorescent dye in their membrane and the receiver with a red one. And when we add the ionophore, you can see that the intracellular fluorescence of the receiver goes up. But does this depend on light? And here we can see the experimental result. You can appreciate that under blue light, there's a much higher signal from the receiver in the receiver cells than in the dark. So we are able to control the communication efficiency using light. So this is the scenario if you have just one sender and one receiver, but in cells, the story is usually more complex, and there can be one sender but multiple receivers. So how would one regulate who is talking to who? And here again, one can use distance as a regulator, and depending on which receiver is in signaling range, the sender can talk to another partner. So to realize this with synthetic cells, we now use a slightly different system called proteinosomes. So these are now protein-based vesicles, and they have a porous membrane. And the surfaces of these proteinosomes, we developed the chemistry to functionalize them with proteins. So in this three population mixture, we have one sender cell, and the sender has a protein called islet and phi B, and another protein called phi B on its surface. And then we mixed it with two potential receiver cells. When we eliminated this mixture under red light, we can see that the sender interacts with the receiver 1 population, which has a protein called PIF6 on its surface. PIF6 and Phi-B bind to each other under red light, and this brings the sender in proximity with the receiver 1. Then we can also use blue light and thereby trigger the adhesions between the sender and the receiver 2, which has another protein called nano on its surface. And thereby, depending on which light we use, we can induce a different sorting pattern within these communities. So I mentioned that these are proteinosomes and they are membrane permeable. And here we teamed up with the group of Steve Mann and Tom de Greef, and they had developed this DNA-based communications between proteinosomes. So what happens here is that we have a sender proteinosome, and when you add a displacement strand, it leads to the release of a signaling strand, and at the same time you can follow this to an increase in green fluorescence from the sender population. Then if there is a receiver nearby, this signal strand can go to the receiver, and there it causes a second DNA displacement reaction and thereby releases a quencher strand, and the receiver fluorescence in the red channel goes up. So we did this with our photo-switchable adhesions, and you can appreciate that in the dark, the sender fluorescence goes up, but the receiver fluorescence does not. However, if we induce then with light adhesions between the sender and the receiver, you can appreciate that first the green fluorescence goes up, and then subsequently the fluorescence of the receiver in the red channel goes up. And you can quantify this, of course, also over time. So this was in the receiver 1 population. We could also do the same thing using the blue light dependent adhesions, so that we could couple the blue light dependent adhesions to signaling between the sender and the receiver population, and it works exactly the same. The exciting thing happens when you then mix all 3 populations. So you have 1 sender and 2 potential receivers. In the dark, you can appreciate that only the sender cells light up in green, but the receiver 1 population circled in red and the receiver 2 population shown in blue do not light up in the red channel. Then if you do the same experiment under red light, you can appreciate that the senders light up in green and the receiver 1 population lights up in red. But the receiver 2 population remains blue, indicating that it has not perceived the signal. And then we can, of course, do it the other way around. We can illuminate the same population under blue light, and now we can induce proximity between the sender and the receiver 2 population, and you can appreciate that the receiver 2 population lights up in purple, showing that it has received the signal. So you can analyze, of course, this population level behavior, and you can you can recapitulate what you see in the pictures. In the dark, there is a population of non-responders, so they haven't perceived the signal. On the red light, the receiver 1 population does respond, but not the receiver 2 population. And on the blue light, it's the other way around. The receiver 2 responds to the signal, and the receiver 1 remains silent. So this is a 3-membered population, but there's not just multiple cells, but there can also be multiple signals in an environment, and this should lead to different type of reactions. And here we thought of how can one induce multiple reactions within a synthetic cell, especially where control of transport is a major challenge. So here again, we are back to the giant unilamellar vesicles. So we have lipid-based membranes, and these are impermeable to polar molecules. Yet we wanted to find a way to induce different type of reactions when we induce the transport of different molecules. And one way biology is doing this is to use transporters that are selective for different molecules. This is not that easy to do in a cell-free system because membrane transporters are notoriously difficult to express, although there is great progress being made. But we went for an easier way and used metal ion ionophores. And these ionophores can be very specific for different metals. So here we have a vesicle. Again, the membrane is labeled in red, and it's loaded with a fluorescent dye. And when nickel gets inside the fluorescent, it's quenched. So now we have here 3 potential ionophores, ionophore A, B, and C. And you can appreciate that only ionophore A induces this quenching in fluorescence, indicating the transport of nickel ion across the membrane. Then we can use the other ionophores to actually induce transport of different metal ions. So the ionophore B, as you can see, selectively transports copper, and the ionophore C selectively transports calcium. Here you should just note that initially the fluorescence is off, and upon transport of calcium, there's an increase in fluorescence. And if you analyze this, you can see that each ionophore only transports its cognate metal ion, but not the others. So metal ion transport is great, but what do you do with that? You want to couple this to function. And for this, we thought of using different metalloenzymes, which we first encapsulated in their APO form. So here we have encapsulated among the other enzymes apo-urease, which is a nickel-dependent enzyme. And when we induce the transport of nickel ions with ionophore A, you can appreciate that the fluorescence in these vesicles goes up. And this is because the urease is activated and it converts urea into ammonia, and this leads to an increase in pH. For ionophore B, we coupled the activity of copper transport to a galactose oxidase. So galactose oxidase is a copper-dependent enzyme, and it can oxidize galactose and thereby generate hydrogen peroxide. And we can follow this again as an increase in the fluorescence using the Amplex red reagent. And for calcium transport, we coupled this to the activity of a phospholipase. So phospholipases are enzymes that can cleave one of the fatty acid chains of phospholipids, and thereby when you activate a phospholipase, you can lyse your vesicles. So we could couple the transport of each metal ion to the activation of a specific enzymatic pathway. So we thought, okay, can we also do this sequentially? And first as A and then another transport a second ion. And actually here, it was a little bit of a surprise. When we first add, for example, ionophore A, we could see the transport of nickel. But if we first added ionophore B or C and then A, the transport no longer worked. So the presence of earlier ionophore seems to suppress the transport of nickel ions. And this is independent of which ionophore you add, or if you add 1 or 2 ionophores that are normally not transporting A. So you see that the addition of prior ionophores suppresses metal ion transport of the later cognate ion. And this was what it was general, so not just for the ionophore A in the case of nickel transport, but this was the same story for ionophore B or C, that the addition of non-cognate ionophores caused suppression in the metal ion transport. So here we see a negative feedback on the transport of metal ions. And we also have looked into the background of this, and it seems to do with how these ionophores move within the membrane. So here with through MD simulations in collaboration with my colleague Andreas Heuer, we could see that the ionophore flips between the 2 leaflets when it's alone in the membrane. But if we add more and more ionophores, they seem to cluster within the membrane, and this decreases the mobility of the ionophores and thereby its ability to transport metal ions. And we can also see this as see that this effect gets more and more important the higher the ionophore concentration is. And this is also the case if we have different types of ionophores, they still seem to cluster within the membrane, and that leads to suppression of metal ion transport later on. So having this adaptivity, we thought that, okay, now we can basically control which enzymatic pathways we activate depending on the sequence in which we add the ionophores. So here we first have added ionophore A, and we can activate the urease. But then when we add ionophore B, in the second step, you can appreciate that we no longer activate the oxidase. And then third, we added ionophore C, which activates the lipase, and the vesicles no longer lysed. And so now we can also, of course, switch this around. We first again add ionophore A and then C, and we could see that the urease no longer lyse, but they seem to get leaky because maybe some phospholipids are still being cleaved. And then when we add the ionophore B, again, we don't see any activation of the oxidase pathway. Or we can do it the other way around. We can first add the ionophore B, activate the oxidase, and then if we add ionophore A, we no longer see activation in the urease, and the same thing for ionophore C. Or the fourth option would be to first add ionophore B and then C, and you again see that you have a little bit of background activity in the lipase, but very, and at the same time, you seem to get some activation of the urease, and we think that this is because of some leakiness of the nickel ions when the lipid membrane is no longer completely intact. So we think that this could be reminiscent of a pluripotent synthetic cell, and depending in which order it encounters different inputs, it differentiates towards different fates. So that we have the first one is the one which basically converts the pluripotent into a multipotent system. The second ionophore seems to be still able to activate the enzymes to some extent at the low level, but then the third ionophore no longer changes the activity at all. What else? Well, we don't just want to control the activation of enzymes, but of course, something that would be even, but also be of great interest is to control the production of proteins and control in vitro transcription translation, which is something that is widely used in the context of synthetic cells. So how can we control when and where proteins are produced in synthetic cells? And of course, the answer is to control these events with light. And this project started off with a very interesting observation that we had with one of our photoswitchable proteins called BCL4. And this protein is different from all the other ones that I told you about. It binds, we saw that it binds to DNA under light. So here in the dark, you see that we have a Cy5 labeled DNA. And when we add BCL4 with the mCherry tag, you see that not much happens. But if we incubate these samples under blue light, you see aggregates of DNA that are co-localized with the BCL4 protein. We also confirmed this with fluorescent polarization measurements, and we could see that every time we eliminate this mixture, there is an increase in polarization, indicating that the protein binds to DNA. And every time, this is reversible, when we turn it off, this goes down. And we can do this over multiple cycles. And as a control, we have a mutant of BCL4, which is just lacking one key amino acid. It's a cysteine that is part of the photoreaction. And when we use this mutant BCL4 instead, we see that we don't see this light-dependent binding between DNA and protein. So this was a nice finding, but then would this be enough to control, to inhibit transcription and translation? So we added DNA coding for BCL4 into an in vitro transcription translation reaction with an mCherry reporter. And you can see that in the dark, we have a rather unspectacular profile. We see production of mCherry over time. And no matter if you add the mutant that is insensitive to light or the mutant or the BCL4 protein. But interesting things happen when we do the same experiment under blue light, and you can see that we completely inhibit mCherry production in this scenario. And so we seem to globally shut down the protein production in this scenario, and we have looked into this mechanistically, and it seems to be that both transcription and translation are globally inhibited with BCL4 under blue light. Well, this now allows us also to control when protein production is activated. So we can first start off the reaction in the dark and initiate protein production, and then stop it when we start elimination. And you can see that then the protein production line goes down, flattens. Or we can do it the other way around. We can first inhibit our reaction, and then when we want to initiate protein production, we can stop elimination, and then we see mCherry production. So it works in both directions. But then why not encapsulate this into lipid vesicles? And so we could see that in the dark, we can have protein production, like here the production of mCherry. But if we incubate these synthetic cells, these liposomes under blue light, we see no protein production whatsoever. We can also use this to control the synthesis of a functional protein, such as alpha-hemolysin. Alpha-hemolysin, as many of you probably know, is a pore-forming protein. And so you can control the release of small molecules from the liposomes using the expression of alpha-hemolysin. You can see that in the dark, alpha-hemolysin is being produced, and so a fluorescent dye that we loaded into these liposomes is leaking out over time. But if we do the same experiment under blue light, you can see that the red fluorescence stays within the vesicles. This worked so well in synthetic cells, so we said, well, what would it do in living cells? So here we looked at the growth of bacteria under blue light, and we saw that the protein is, the cells are growing under dark. But if we culture the very same cells under blue light, the cells no longer seem to grow as fast. And this is not phototoxicity, because if we do the same experiment with mutant protein under light, these cells grow just fine. And we can actually use this to control bacterial growth on a logarithmic scale and with high temporal control. So we can have phases of cell growth. And then when we start elimination, we stop cell growth. And then when we stop eliminating again, the cells grow, and so on. So we can control this growth and arrest of growth reversibly. And if you're not, so seeing is always believing. So here's the same thing under the microscope. So here we have E. coli growing on an agar plate, and you can appreciate that in the dark, these bacteria are growing and dividing, whereas in the blue area, there is no cell growth. You can even see that the bacteria from the dark area overgrow and push into the blue illuminated area. Yeah, finally, I talked about a lot of chemical signals, but could we even use light as a signal to communicate information between synthetic cells? And this actually is quite inspired by many fiber optic communications. So light, of course, is a very attractive signal because it would travel extremely fast. And actually, there's many macroscopic examples where light is used for communication in biology. Yet, at the cellular scale, it has not been described. However, there would be obvious advantages. Of course, if communication was based on a light signal, it would propagate extremely fast at the speed of light, and it would be independent of diffusion. It would be able to cross transparent membranes, such as lipid membranes, without the need for a transporter. And it would be a completely independent channel of chemical signaling. It would be highly specific because light production as well as light response is not something that is very common. And we could use this, introduce this with dedicated molecules. And if we understand how to do this, we could code even different types of information using different colors or intensities or frequencies of light. And so this has not been described in biology, but if we approach this from a retrosynthetic perspective, this is actually not a difficult problem. What would you need? You would need intracellular light production, and you would need extracellular light responsive element to mediate this. If you break it down, okay, intracellular light production, one can just encapsulate a light-producing enzyme into a vesicle in a sender cell. And so we did, and we encapsulated a vanilla luciferase within a giant vesicle. And then when we add the substrate, we can see that we can generate high photon counts. And these decay, of course, as a substrate is consumed, but we can actually re-initiate light production when we add new substrate. How about the light responsive element? Here we looked at the spectrum of our blue light emission from the luminescence, which is shown here in orange. And we saw that it matches very well with the absorption spectrum of our photoswitchable proteins, such as islet. And so we have here potential light responsive elements with good spectral overlap. And then we asked if such light production within a vesicle could activate these proteins. And so here islet is immobilized on the surface of a vesicle, and on solution we have a fluorescently tagged binding partner, and we can see that it's recruited to the membrane as an increase here in green fluorescence. And so there's an initial increase. And later on, when substrate is consumed, this is reversible and goes down again. So we then can, of course, use this to, again, mediate adhesions between sender and receiver cells. And we can see that when the sender cells produce luminescence, we get, again, aggregates of sender and receiver cells. And this is not happening in the dark, and it also happens under blue light, which we use as a positive control. And now we were inspired by the anglerfish. So what does the anglerfish do? It produces light to lure this fish. And when the fish, the prey, comes into proximity, of course, then the anglerfish goes for the kill. So could we do this with synthetic cell at the microscale? And so I already showed you that we can generate luminescence and use this to trigger adhesion. Then we coupled these adhesions to a secondary chemical signal that would only be transferred from the sender to the receiver upon adhesions formation. And for this, we again introduced these alpha hemolysine pores that were initially blocked. But in the adhesion area, the blocker is displaced so that the pores open, and then you can transfer a small molecule from the sender to the receiver side. And here, this small molecule was again a calcium ion, and we used this calcium ion to activate the apophospholipase that was introduced into the prey cell. Here is the experiment without the lipase. We can see that we have a contact-dependent transfer of calcium ions from the sender, shown in green, to the receivers, shown in red. And you can appreciate the transfer of the calcium ions when the sender and receivers are in contact, but not to receivers that are far away. And here's then how it looks under the microscope. You have here the predator in green and the prey in red. They are in contact, and upon transfer of the calcium ion, you can see that the prey lyses. As we have a molecularly defined system, we can then really ask if each component is really doing what we intended it to do. So we can mix equal numbers of prey and predator GUVs, and we can see that only the prey GUVs lyse selectively, not the predators. When the predator is luminescence, when the prey contains apophospholipase, and the predator has the calcium ion that activates the lipase in the prey upon transfer. So we can really generate this type of hunting scheme in synthetic cell communities. The final point I would like to make is that, of course, conversations should be regulated. You have a conversation, but after some time you run out of signal, and then there needs to be a way to end the conversation and leave. And we also replicated this in synthetic cells. And the way to do it is to couple the proximity of sender and receivers to the signal production. And here, for this purpose, we use hydrogen peroxide as a signal. which can be produced in a sender and transferred to a receiver cell. But now the question is, how do you couple this to adhesion? And for this, we use a different type of luminescence. We use chemiluminescence to generate, to photoactivate our adhesions. And so we can use a hydrogen peroxide for a second purpose. It can react with luminol and produce light, and this light can then activate the adhesions. And you can appreciate that these light production will only happen as long as there is hydrogen peroxide, so signal production in the sender. And once the signal goes down, then these adhesions are reversible and sender and receiver can detach. So first of all, to show here under chemiluminescence, we can trigger adhesions between sender and receivers, and we can also transfer the hydrogen peroxide signal from the sender to the receiver, which we can see here with a fluorescent dye, and you can appreciate that the receivers light up with a cyan color, showing that they have gotten the hydrogen peroxide signal. And then we can, of course, tune for how long we produce luminescence, and this depends, of course, on how much enzyme we encapsulate. And we can see that this light production also correlates with the contact time between sender and receiver. You can see that we first form aggregates between sender and receivers, but over time, these reverse as the light production goes down again. Finally, these conversations should not be one way, but there should always be a response to the initial signal. And also to do this, we have used these same synthetic cell pairs where we had a hydrogen peroxide signal from the sender to the receiver, but then we got calcium as a response from the initial receiver. And to get this calcium response, we used alpha hemolysine pores that were initially only implemented on the sender, and only upon contact with the receiver, these pores opened and the calcium signal was transferred. So here you can see that only sender and receivers that are in close proximity have this two-way conversation, which is visible by the hydrogen peroxide center shown in cyan, and the calcium signal on the sender cell shown in violet. So yeah, I think we have gotten quite far with our synthetic cells in terms of communication. So we can control how efficiently they communicate with distance. We can control how they communicate within mixtures and between multiple partners. We can show some adaptive behavior in terms of the communication behavior. We can use light as a signal between synthetic cells. We can regulate in vitro transcription and translation with light. And we can introduce self-regulation and two-way communication. A final point is that we use similar photoswitchable adhesion molecules also on bacteria and mammalian cells, and this is a way going forward to produce hybrids of these. And just initial work that we have already done is here between cargo molecules and bacteria, and these bacteria bots, as we call them, can assemble under red light, and then these bacteria bots can carry their cargo to some distance, navigate complex environments, and then when we illuminate with far red illumination, they can drop their cargo. So we think that if by building in communication between these bacteria and their cargo, we can generate smarter and autonomous bacteria bots. So yeah, I hope that the examples that I've given you have convinced you that photoswitchable proteins would be a valuable addition to our synthetic cell toolbox. And with this, I come to the most important slide. So I have to thank my team who has done all of this work. And it's a very good mix from people from all over the world across different disciplines, ranging between chemistry and biology. I also would like to thank my many collaborators who have enabled this work. And of course, you for having me here, and I'll be happy to take any questions. Thank you.
Thank you so much. Thank you for a great talk. There are several questions in chat. Can you see the chat?
Yeah.
If you could read the question out loud before answering.
Yeah, so here's a question from Tomo Fujima. How do you regulate the adhesion attachment point number? I would imagine too many would pancake the vesicles to the surface and not enough would cause weak adhesions. You're absolutely right. So this is the painful optimization that the PhD students go through. And here it's very important to control the functionalization density of these proteins on the surface and on the vesicles. But this you can do actually in these systems quite well. It's much easier than controlling expression levels in cells, for example. You just control the number of NTA groups that you put into your lipid mixture or that you put on your surface. And maybe I should also mention we have different photoswitchable protein pairs that we use for this purpose. Some of them are stronger than others. Some reverse faster or slower in the dark. And these are other ways we can optimize our systems. Okay, then the second question comes from Anna Maria. Does membrane composition influence ionophore performance in your pluripotent cell experiments? What is the membrane you used in most of the work? Great experiments, thank you for presenting. Yeah, so membrane composition is certainly very important. In the case of these ionophore experiments, we try to keep the membrane composition extremely simple. And it's just DOPC as far as I remember. For some of the experiments, we add a little bit of negatively charged lipids to have some electrostatic repulsion between the vesicles so that we don't have too much background aggregation. And yeah, so it goes case by case. Sometimes, especially when we combine vesicles with living cells, it's good to add some cholesterol because that stabilizes the membrane and avoids fusion, for example, with native cell membranes. So I think, yeah, one has to really go about this case by case. So yeah, Bertha, vesicle solutions are usually pretty turbid. Do you see that as slowing down or introducing noise to your light controlled systems, or do you have always to deal with scattering with liposome particles? You're absolutely right. Scattering is, of course, a big limitation of light controlled systems. In our case, we have, I think, very low densities of vesicles, so that we circumvent this problem. I think if we were in more crowded systems, scattering would be a bigger problem. And we do see this in cases where we have more concentrated bacteria or cell samples. That's where we usually then have these problems. But that usually, of course, that is just a problem for spatial resolution, and you still keep the temporal resolution, which is always there. But light penetration, of course, is also there, a problem if it gets too crowded. Avery, amazing talk, thank you. Haley McKinsey, did you ever try adhesions to anything other than flat surfaces like hydrogel, embedding the synthetic cells in some matrices? Very good question. Until now, we have only done it with flat surfaces, just because it gives us very easy control of this functionalization, and it's easier for microscopy. But yeah, there is also some photoswitchable hydrogels based on such proteins, and I think that would be a really nice way to going forward. Yeah, a question from Ahmed. Great talk. In the bidirectional communication experiments, you mentioned that there is no alpha hemolysin in the receiver cells initially. I was curious if you think that the alpha hemolysin leaked from the sender to the receiver, or perhaps the membrane fused. So we did some controlled experiments, and it's not leaking in the sense that there's alpha hemolysin that dissociates and travels to the solution to the receiver, but it seems to be something that happens on this transfer of alpha hemolysin only happens when the two membranes are in contact with each other. I don't know the exact mechanism behind it, but we think that there might be a flipping mechanism when the two membranes are in close proximity. We did not see fusion of the membranes because as we also have fluorescent dyes in these two membranes, and we did not see flipping between these two channels. yeah, those were all the questions in the chat, but if you have any other questions, please feel free to also ask in person.
I think that's all the questions. I was very happy that someone asked about titrating the adhesion element, because we deal with the same problem, and
Yes.
I'm always curious to hear how people solve that. Is there any way to solve that other than just brute force? Because that's how we do it too. We just do the experiments and see what comes out. Do you have any idea, like, can we predict in advance what molar percentage of the membrane should be the adhesion elements, or it's always experiment?
Well, I think we can by now say that I think what's very important is the KDs of these two protein interactions. And so we have measured this for our islet nano pair. And so we are in the high nanomolar range when we are talking about blue light illumination, and we go to the micromolar range when we are in the dark. So I think if you have insight about the KD, that can give you some predictability of how much you need. So I can tell you with these kind of low or high nanomolar functionalization density around 0.5% to 0.1% is enough. Yeah. But yeah, I think getting KDs is good, but then of course also the systems are studied in very different contexts. And the other thing is of course multivalence. So these switches that we use work very fast, and we don't see clustering at the short time scale. But I can tell you in unpublished data, we have seen clustering of these adhesion molecules when we wait longer, and if we have more mobile membranes. And that, of course, then also suppresses reversibility, which you may or may not want. So I think there's also some value in optimization and a lot of insight to be gained there.
Yeah, that's our experience too. And
yeah.
I'm sad to see someone's dealing with this problem too, but I'm glad we're not the only ones.
Yeah, I think this is like, if once you have a handle, you can really build the things together, but establishing them one by one is hard work.
Yes. All right, if there are no more questions, I want to thank you, Seraphine, again for a fantastic talk.
Thank you for having me.
Thanks everyone else for a great discussion, and have a great week.
You too.
Bye.